Welcome to Veterinary Bacteriology and Mycology. My name is Joe Rubin, and I'm a professor in the Department of Veterinary Microbiology at the University of Saskatchewan, and I've been teaching this course since 2016. The objectives of this course are to prepare you for your clinical years in the veterinary program. So our goal here is to help you build a strong foundation in microbiology, to be able to speculate on the pathogenesis of diseases, to understand how the diagnostic process works, and how, as a practitioner, you can work with the diagnostic lab to both ask the right questions and also then be able to interpret the results that the lab provides for you, to have a foundational understanding of antimicrobials. Now, this isn't a class in pharmacology, so this is meant to really be a primer in antimicrobial chemotherapy, to be able to recognize and prioritize zoonotic risks and ensure a safe working environment for both yourself and your staff. And then finally, to be able to communicate concepts in clinical microbiology to the lay public, as this is really going to be your job as a veterinarian. This course sits in kind of an awkward position between basic microbiology and clinical infectious diseases. We're talking about foundational microbiological concepts through a clinical lens, but this isn't a class in clinical infectious diseases. That will come later in your third year medicine and surgery courses. Now, this course draws heavily on textbook resources. These are some of the books which I find extremely helpful and have really formed the basis for this class. I strongly recommend that as newcomers to the field, you utilize textbooks as your first source of information. Textbooks do a great job of summarizing the most important information and contextualizing it and prioritizing it for you. Digging through the scientific literature can be really exciting and interesting, but it's oftentimes overwhelming at the beginning. So this course draws from both clinical microbiology textbooks as well as relevant medicine textbooks to have some accurate descriptions of diseases. Where there are recent advances or developments in microbiology, I have drawn from the scientific literature, and I would encourage all of you to check out our library website. Here you can do a search of reputable peer-reviewed journals. This is a screenshot of the University of Saskatchewan library website, where you have access to all of the contents we use in this course and so much more. Microbiology is the most dynamic field. The very nature of what we work with makes it an exciting and rapidly evolving area. And I think we've seen that demonstrated quite clearly over the last few years of the pandemic. At the beginning of the pandemic, I actually had the opportunity to do some traveling in Southeast Asia. And so here's some pictures from my trip where I got to experience the pandemic as it unfolded. So in late December 2019, I was at the Chengdu Panda Center in China. Following that, you can see this sign at the Changi Airport in Singapore on February 8th, noting that people who had been to China, mainland China, in the last 14 days would not be admitted. We had the initiation of temperature checks in Singapore and in Kuala Lumpur. And then here on March 4th, 2020, this is the flight board at the Istanbul airport, and you can see cancellations of flights to sort of the initial hotspots of COVID-19. Initially, it was in Italy and Iran. And then finally, this was my first COVID positive test in May of 2022. Having said that, this is not a virology course, so we are going to be talking about bacteria. And one of the most important and impactful emerging issues in bacteriology is that of antimicrobial resistance. This is a topic that we see covered a lot in the media, whether it's about this sort of looming global health crisis and urgent calls for action, threats of zoonotic spread from our companion animals, increased risks of death, or novel solutions to antimicrobial resistance. So while COVID has been the infectious disease on everybody's mind, The slowish burn of the AMR pandemic is likely more damaging over the long term. This is a screenshot of a very famous report, the ONeill Report, which came out in May of 2016. And what the authors did was tried to project into the future to 2050 and say, what will happen if we continue on our current trajectory? If resistance continues to emerge, how is that going to be impacting us? And what the authors found, or what they projected, is that by 2050, we will have over 10 million deaths attributable directly to antimicrobial resistance, which is more people than are currently dying of cancer. So it's certainly a serious threat. It's one that promises to have a very dramatic and profound human impact. More recently, this study published in the Lancet actually looked at what the current global burden of antimicrobial resistance is as of 2019. And what they found is that we are already seeing over 1 million deaths annually attributable directly to antimicrobial resistance. So this is a very, very serious and very contemporary threat. In Canada, we have some really good antimicrobial resistance surveillance programs, and these are some screenshot infographics of the CARS report, the Canadian Antimicrobial Resistance Surveillance System 2022 report, where they highlight some of the emerging threats. Two that I've picked here are drug-resistant Neisseria gonorrhea, which is trending up, as are the carbapenemase-producing Enterobacteriales, trending up. This report looks not only at the frequency of antimicrobial resistance, but also how antimicrobials are used. And so in this histogram, what you can see is over time on the x-axis from 2017 to 2021, the amount of antimicrobials used per population in Canada, both the total amount, the amount used in community and in healthcare. And I think what you can see is that there is a trend of downward antimicrobial use in people. This is a very, very hot topic, and we're certainly trying our best to reduce particularly unnecessary prescribing. We know that unnecessary prescribing can amount to a very potent selection pressure for antimicrobial resistance. And the ONeill Report highlighted this. So of the 40 million people who are given antimicrobials for respiratory tract infections just in the United States, 27 million of those are unnecessary. So a lot of drug use that we can eliminate. There's also an increasing emphasis on not just eliminating inappropriate antimicrobial use, but reducing the duration of therapy. Fewer drugs in the system, less selection pressure for antimicrobials, and also a lower risk of adverse drug reactions. The CARS report also looks at antimicrobial use in veterinary species, so it's not just focused on people. And certainly whether we're talking about companion animals, dogs and cats, livestock, so animals that enter the food supply, drug use is coming under increasing scrutiny. And so as veterinarians, we need to be really mindful how we're using these products and ensure that they're only being used when absolutely necessary so that we reduce the selection pressure for resistance. So with all of these threats that are emerging, whether it is COVID, whether it's antimicrobial resistance or other issues in bacteriology, how are we keeping up? Microbes are a really formidable opponent, and we need some really powerful tools in order to combat these emerging threats in infectious diseases. And our ability to keep up really relies on something that's perhaps a little bit unexpected, and that is, in fact, computing power. So this is a figure that demonstrates Moore's law, which is really more of an observed association. Moore's law states or describes that the number of transistors that can be incorporated into a dense integrated circuit doubles approximately every 2 years. So essentially the computing power that we have. And this is a relationship that's held true from the 1970s through present day. So you can see on the x-axis here, year, and then the number of transistors on a logarithmic scale on the y-axis. Certainly when I think back to the first computer that I had, this compact portable back in the mid-1980s, it wasn't terribly powerful, and it certainly wasn't very portable either. To contextualize these rapid advances, we can look at the computational capacity of the fastest supercomputers. So the number of operations that can be performed per second, going from under 1,000 in the early 1990s up to 1 billion today. And then also the historical costs of computer memory and storage, which you can see reducing very, very, very quickly. We're now down to under $100 per terabyte, while in the 1950s, we were in the billions of dollars per terabyte. So what could this possibly have to do with infectious diseases? Well, it all comes down to genomics. Back in the early 2000s, there were these competing projects to sequence the human genome, one the publicly funded project and one by the Venter Institute, very famously published sort of these competing articles in Science and Nature. The first human genome took 13 years to sequence. It took warehouses full of equipment and cost billions of dollars, $2.7 billion. Today, we could sequence a human genome for approximately $1,000 in a single day on a single machine. If we relate this back to Moore's law, what you can see is that our rate of advance in the genomics field has actually outstripped it. So this is 2.5 million times less expensive, 5,000 times faster in only 20 years. If we look at these advances in the context of actual data that's been generated, I think this paints a really clear picture. So these are some figures that I've taken from the National Center for Biotechnology Information, NCBI, which runs Genbank, that's a repository for genomic data. And what we can see is that there have been logarithmic increases in the quantity of sequence data that's been produced. So in both figures here on the x-axis, we have years from the early 1980s up through present. And then on the y-axis, we have the number of base pairs worth of data. So those single A's, T's, G's, and C's that make up our genomes and our bacterial genomes on the y-axis. If we look at this red line here, this is whole genome sequencing. And I think what you can appreciate is that in the early 2000s, this really started to take off. This was due to the introduction of next generation sequencing technologies, which really revolutionized the genomic sphere. If we look at this data with a little more granularity, what you can see is just how rapidly it's evolved. So in 1982, with the third release of Genbank sequence data, you can see that we had just over 600,000 base pairs in 606 individual sequences. And as we move through time to the turn of the millennium, you can see that increasing quite rapidly, and that continues. That trend continues. So here was the first time I taught this course back in 2016. Here's the onset of the pandemic, and here's where we are today. If we look at the number of base pairs we have here, we're now at over 21 trillion from 2.6 billion genomes. And this is only the data that's actually been deposited into Genbank. It's actually becoming so routine now to use these sequencing technologies that the data isn't always even uploaded. So we're talking absolutely astronomical numbers here. There's between 100 and 400 billion stars in the Milky Way, so several hundred times more nucleotides in Genbank. So this is really, really big data. In fact, it's so much data that it can actually be really hard to handle. How do we actually make something useful of all of this information? You know, we're really getting hit with a wave of data that needs to be interpreted. Back just over 10 years ago, this paper came out sort of prophesizing what would happen when we had 10,000 bacterial genomes published. 10,000 genomes sounds kind of quaint now. You can see on this figure from NCBI, the number of bacterial genomes here in blue has increased really exponentially. And in 2016, when I first taught this course, there were over 72,000 genomes deposited in Genbank. Earlier this year, we were closer to 600,000. So it's continued to grow, it's continued to develop, and this wave of data is continuing.